All right, let's go ahead and get started. Today is Wednesday, September 30th. We are on to our third section of the class, so we are rapidly racing through the material, and that is the skeletal system. We will start today primarily focusing on the anatomy and histology of our organs that are the bones, and then in groups. So this will be really the first time up to this point, what we've been talking about in lecture, what we've been talking about in lab, pretty much hand in hand. In this part moving forward, hold on, there we go. On this point moving forward, we're going to see a little bit of a divergence. We're going to be, obviously today we're going to be talking anatomy, but then the rest of the time we are going to be focusing on the physiology of the skeletal system during lecture. And then lab will be focusing on the massive amount of anatomy you are going to be responsible for. As I said, rather than me standing up here and holding up the bones for you and going, all right, this is the start button and this is that button and so on and so forth as we go through it, to make it more engaging, I'm going to be breaking you into groups and you will be presenting the bones and bone features that are on your list. We will form the groups today. I have provided you with all the resources for you to be successful with that, but you're welcome to be creative and use other resources if you would like in doing that. And those presentations will start on Monday. And so we'll continue them. Next week, we'll do the axial skeleton. The following week, we'll do the appendicular skeleton. So you'll be presenting twice. But like I said, it's very low key. You're not getting graded on your presentation skills. It is a graded assignment, but really it's just you're getting points for being active in the learning process. And so we'll talk about that more after the lecture today. There are 4 assignments for this section. Unit review 7, 8, and 9, and you know what to do with that. There are both check your recall and check your understanding, 2 parts of all of those. Both of those parts are due, and they're due at the beginning of class, and they are graded for completeness. I'm looking for time and effort. There is a 4th assignment, and that 4th assignment is the 30-point skeletal review. This assignment is worth 30 points, and it is graded for correctness. What that means is, unlike the unit reviews, which I really want to be learning activities for you, if you get a question wrong, as long as you put time and effort into it, you get the points. That is not the case here. If you get a wrong answer on the skeletal review, you lose points. Now, I know people see 30 points skeletal review, people see graded for correctness, and people want to start working on it right away. But if you've noticed, it's not available yet, and that is on purpose. Because what we are going to be doing in this class over the next couple of weeks is focusing on all of the minutiae of detail. You are going to be staring at individual bones and learning about all of the pieces of all of the individual bones. But what we need to remember is these bones fit together to form our entire skeleton. They work together. And so one of the big keys to the skeletal review is that for bone 1, not only do you need to know its bits and pieces, and for bone 2, not only do you need to know its bits and pieces, but you need to know how bone 1 and bone 2 go together. So on the exam, for instance, I may show you a bone feature of a particular bone, and I could ask you what to identify what it is, but I could also ask you what it articulates with, what other bone it is. And if you don't know this bone and these bone features, and if you don't know this bone and these bone features, then you're not going to know how to put them together. Could you, right now, today, start using brute force to try to go through these things and come up with the right answers for this? Yes, but it would not be a good use of your time. Instead, for the next week or so, I want you focusing on learning the individual pieces. And then as you start learning the individual pieces, it'll be much easier and much more meaningful for you to see how they go together. So I purposely have not let the skeletal review be available yet, because I don't want you looking at it. I don't want you thinking about it. I don't want you focusing on it. I want you focusing on the current material. And as you learn that current material, it'll make the review much, much easier. So I'm not giving you the review till later. You'll have a full week to be able to work on it before it is due. That is going to be plenty of time. But you are going to be working on it in a roundabout way by learning the bones and bone features now. And then, of course, on the 19th, you have your lab and lecture exam. You have now taken 2 lab exams and 2 lecture exams. You know exactly what to expect from those. The third is going to be the same as the second and the first, which is going to be the same as the fourth and the same as the fifth. So there's no surprises on that. You know exactly what to expect. All right. Questions on the game plan?
No.
Excellent, excellent, excellent. All right, then let's go ahead and dive into the new material. Our skeletal system. Got my windows where they belong. Perfect, excellent. All right. So I don't remember if we talked about this before, but skeleton comes from the Greek word skeletos, which means dried up. Because usually when we see a skeleton, right, we see that dried up a bone sitting out on somebody's tabletop or something along those lines. However, as we have talked about, bone is very well vascularized and it is an incredibly dynamic tissue. As we talked about, someone breaks their femur, right? And in 6 to 8 weeks, it's going to be healed and they're going to be as good as new as a result of that. So it is incredibly dynamic with a whole lot of stuff going on. The skeletal system is indeed an organ system, and so it needs organs. And are there organs in the skeletal system? What do you think?
Yes.
The bones.
And what would those be? The bones, absolutely. Are the bones really an organ, though? What was our criteria for an organ when we were looking at that hierarchy of organization?
Made of tissues.
Tissues, plural, right? That's the key. It is made up of tissues, plural, working together for some function. Is a bone made up of more than one tissue?
No.
Well, I've got a no and I've got a yes. Who's right?
Yes.
Yes.
Right. So give me an example. What tissues, plural, might it be made up of?
Blood, maybe?
Yeah, there's going to be blood vessels inside of it, absolutely. Red bone marrow, which is made up of reticular connective tissue, right? It's got cartilage at the end, as we've talked about. There are nerves that pass through it, which is why it hurts when it breaks. Now, why we get confused is because the majority of the bone is made up of that bone connective tissue, right? But again, that's one of the reasons why when we were talking about connective tissues, we talked about sometimes using the term osseous tissue instead, so that we don't get confused between bone organ and bone connective tissue. But yeah, absolutely, it is made up of more than one tissue, many tissues inside of it. So it does indeed meet the criteria of being an organ. And just out of curiosity, how many bones do you have?
206.
206 bones? I like what Alina wrote a lot better. You have 206 named bones. Every single person in this class has far more than 206 bones in their body. More than the difference is that many of these bones are tiny bones, individual bones that are different places in you than they are in me, than they are in somebody else. And so if I have a bone in my skull right here, a tiny little bone here, and someone else has it here, and someone else has it there, right? If there are 3 different locations where you can find that, then we can't give it a name like Sally or some of the other names that they give to bones. And so there are plenty of bones that aren't named. But you are right, 206 named bones. And of course, how many of those 206 named bones are you going to need to know for this exam?
All of them.
All of them, absolutely. You need to know all of them. And if they are a paired bone, and what do I mean by a paired bone?
Left and right.
Yeah, if there's a left and right, then you need to, if you have 2 of them, then you need to be able to distinguish left and right. This usually is one of the funnest sections in this class. Everybody loves the bones, right? But that's because we get to hold these things in our hand, gnaw on the ends of them, right? Stick them in our ears, do things like that with them. You're not going to have that opportunity anymore. So instead, you get to look at pictures of bones and animations of bones, and it's not going to be the same, especially in trying to be able to differentiate left and right. It's going to be a lot more challenging. So this section is a lot less fun and a lot more challenging than it normally is. So it's going to take a lot of individual work. Luckily, we have a lot of resources to help you to do that. And so there are ways that we're going to get around that and try to make it successful. But I will warn you that this is one of the more challenging sections in this format, because the bones are normally such a tactile section in this class. And unfortunately, right, unless you have some of these hidden in your closet, you're not going to be able to manipulate them in your hands. All right, but absolutely, 206 named bones, and we'll learn all of them. And of course, the study of bones is the field of osteology. All righty. So, like all organ systems, our skeletal system has functions. And there are 6 main functions of the skeletal system. We've talked about kind of the big 3. Nope, seeing as we are already 10 minutes into the lecture, I'm going to go out on a limb and guess that there isn't a daily quiz today. Unless it's at the end of lecture, which can always happen. So give me an idea of what the 6 possible functions for the skeletal system could be. Give me one of the obvious ones. Give me an example. Support. I love that. Absolutely. Right, support. We talked about how our skeletal system provides our vertical axis to the body. It allows for movement. They are the levers that our muscles pull on to be able to provide that movement through space. And what's the other big one?
Protection.
Protection, absolutely. Perfect. Those are by far the big 3, right? And I like that we did it in that order as well, because often in many classes, protection is the first thing that people say. And while it is absolutely true, our skeletal system provides protection, right? Our old noggin protects the brain. We have these ribs that protect our heart and our lungs and our liver and things along those lines. But how many bones do I have in my hand?
A lot.
A lot. Okay, I'll let you get alone with a lot this week. Next week, when we move on to the appendicular skeleton, we may actually have to give a number to that. But I'll give you a lot today. Absolutely. There are a lot in this hand. And are any of them providing protection? No. They give the structure, they allow for movement, they do all of those things, but they're not providing protection. But absolutely, those are the big 3. But if I look at the slide here, it says that there are 6, so there must be more. What else does our skeletal system do for us?
What about growth?
I'm sorry?
Growth?
True. So you are correct. That is definitely one of the functions of the skeletal system. I mean, that is a function of it, being able to be dynamic, being able to grow. That is definitely an important component of it, but I wouldn't necessarily say it's a function of it, although I do like that. I do like growth. We'll throw that on there because I do like that, but it's not one of the 6 we were talking about, but I do like that a lot. There you go. I like the making of the blood. Right? All of the formed elements of our blood are made in our skeletal system. Where? Where do we make all of the formed elements?
Bone marrow.
In the bone marrow. Can we be more specific though? Ah, there we go. Red bone marrow.
Red bone marrow.
Yeah. Excellent. I don't remember if we talked about this when we were talking about, I don't think we did when we were talking about blood as a connective tissue. There is a pluripotent stem cell. Remind me again what pluripotent stem cell means?
More than one.
More than one.
Exactly. It is a cell that is undifferentiated, divides to produce lots of cells, and it become more than one thing. And it forms all of the cells of our blood. The red blood cells, the white blood cells, and the platelets, right? And it is called a hemocytoblast. So that hemocytoblast is responsible for making all of the formed elements of our blood. Of course, making the blood is a good way of describing it, but of course, as we know, anatomists hate us since they like to give fancy names for these things. And the fancy name for this is hematopoiesis. There we go. Hematopoiesis. Hematopoiesis is the process of making, and let's be more specific, making the formed elements of blood in the red bone marrow. Excellent. And that's good. But again, remember also, Allison, being the smart, sophisticated students that you guys are, does it really matter for the exam that you pronounce it correctly? One of the things that I will say for myself personally is I am not typically a good speller. Exactly, because you have to type it out. Yeah, I'm not a good speller. So as I was a student learning all of these myself, I often missaid things based on what they looked like. I pronounced things the way they looked in my head, so that would help me to spell them. One of my biggest challenges when I became a teacher was I had to relearn how to say all these things the right way because I've been saying them wrong in my head for decades. So as long, again, being a smart, sophisticated student, I don't care if you pronounce it correctly as long as you spell it correctly. All right, so I think I saw the other answers as they raced by, but we've moved a little past it. So give me another function. We have 4, there are 2.
Mineral storage.
Mineral storage, excellent. Mineral storage, like what?
Calcium.
Calcium, absolutely. Calcium is the big one. As we keep talking about, calcium is what makes cells do wonky things. And something like 99% of all of the calcium in the body is stored within our bones. Right? Vitally, vitally important. However, right, we do have to put a bit of an asterisk by this. Because as we've mentioned, calcium makes cells do wonky things, right? If you want to contract a muscle, you need calcium. If you're a gland that wants to release a secretion, you need calcium. If you're a neuron wanting to produce an action potential and spread a signal from one cell to the other, you need calcium. So it is vitally important we keep the appropriate amount of calcium in our blood. Now, I know this isn't an issue for anybody here in this class, because I know you all had a nice big square breakfast that included a big, large glass of milk with it. But there are some people who will have a Diet Coke and a donut for breakfast. Are they necessarily getting the calcium that they need into their body?
No.
No, exactly. And so part of the problem with that is if the body has to make a choice between having the right amount of calcium in the blood and having high levels of calcium in the bone, who's going to win?
The calcium.
The calcium in the blood. And so if you're not getting enough calcium from your diet, we actually go to the bones and pull it out of the bones, weakening the bones in that process. So definitely is where we store calcium, but if we need it, the body has no problem going there and getting it. But it's not just calcium that is stored there. There's phosphates, magnesium. I mean, all sorts of different minerals and other electrolytes are stored in the bone. And so then the last question I have is where? Where in the bone is all of these components stored?
And the yellow bone marrow?
No, not the yellow bone marrow.
In the matrix?
Although we will talk about yellow bone marrow in a second. There you go, exactly. In the matrix, right? This is stored in the matrix of the bone connective tissue, right? So that matrix of the bone connective tissue is where we store the minerals. You guys have mentioned yellow bone marrow a couple of times, and that is an important substance as well. What's the big difference between red bone marrow and yellow bone marrow?
Yellow does not make black.
Okay, you're right. Yellow doesn't make the bone cells. So what
It makes
nutrients?
Nutrients.
True, and primarily I think Daniel's got it right. It is lipid storage. We store lipids in there, fat. Yellow bone marrow is basically red bone marrow that has accumulated a massive number of adipocytes. And those adipocytes basically store as triglycerides, store fat. Why? Why do we need to store fat inside the bone?
For cushion.
True, cushion is a great answer. That's normally what we think of, right? We think of that cushion that that fat provides for parts of our body. But if we're putting it on the inside, is it really providing cushion? Energy, definitely. All adipose is providing energy. Is there any other reason you might put it on the inside of our bones? Is it on the inside of all of our bones?
No.
Probably not.
Which bones primarily?
Long bones.
Long bones, absolutely. Big bones, right? And there you go. I like Alina's got it. It helps with the weight distribution, balancing of the bones, balancing of the body, right? Again, we're quite a ways from January, but usually the beginning of January, right, everybody's worried about fitness and getting their New Year's resolutions and all those. And so one of the things that they love to do is on the infomercials, they love to show you a pound of muscle and a pound of fat. And are they always the same size?
No.
No, the pound of fat is way, way bigger because fat is a very light material. And so by taking these big, huge, massive bones and filling the inside of them with fat, yes, we get storage of energy. We just said bones are very, very dynamic, and having that energy stored there is useful and helpful. But it also helps with weight distribution. It also helps us to lighten the skeleton so that it's easier to move, right? It's not so exhausting to provide that support. So absolutely. Excellent. Perfect. So there you go. Those are our 6 functions, right? Support, protection, movement, hematopoiesis, mineral homeostasis, and triglyceride storage. Excellent. So what I would say is that, again, we haven't talked about the endocrine system yet in too much, and we won't get to that till 4:30. One of the cool things about the endocrine system is that we still continue to learn so much about it, almost literally on a day-to-day basis. And we do indeed find more and more that pretty much every cell in the body seems to have some potential to produce chemical signals, hormones, that allow it to communicate with other cells. In the red bone marrow, we do have cells that produce hormones. However, the hormones that are produced there are primarily local hormones, regulating which, because again, we have this issue with our hematopoiesis, right, where we have one cell to make all of the formed elements. So does that mean on Monday it makes red blood cells, and on Tuesday it makes white blood cells, and on Wednesday it makes platelets, it gets to take Thursday off, and then it's back at it again? No. How do we tell that hemocytoblast when it divides what we want these new cells to become? Hormones, absolutely. So there are a fair amount of hormones that are involved in this process. Hormones produced in other areas control our mineral storage and things along those lines. So there are some hormones that are produced there, but they're primarily local, so they're not affecting other parts of the body in that same way. But yeah, we're finding more and more that almost every cell in the body has some ability to produce some type of chemical signals. All righty. Bone is indeed an organ, but that organ is primarily made up of bone connective tissue. We talked about it briefly when we were talking about our connective tissues, because it is indeed a connective tissue. But we also said we were going to talk a lot more about it when we get to this section. And so here we are. So bone connective tissue, or what we can think of as osseous tissue, like all connective tissues, is made up of 2 main components, cells and matrix. So those are the things we need to talk about. And the first thing we need to talk about is that there are basically 4 cells that are used to form our bone connective tissue. So let's talk about those. The first of these cells is what is known as an osteogenic cell. Osteogenic cells are unipotent stem cells. Someone remind me again what a unipotent stem cell is?
One bone.
Cell.
Exactly. It is a cell that is capable of dividing to produce new cells, but when it divides to produce new cells, it only makes one type of cell. These osteogenic cells come from our mesenchymal stem cells. Remember, our mesenchymal stem cells are the pluripotent stem cells. Oops. That can produce all of our connective tissue cells. So a mesenchymal cell will divide to produce an osteogenic cell. And this osteogenic cell is still a stem cell, but all the cells that it makes can become one thing and one thing only. One of the special things about these osteogenic cells are these are the only bone cells that are capable of division. So our mature osteocytes, when they're hanging out inside the bone matrix, those cells can't divide. Our osteogenic cell is the only bone cell that is capable of division. Now, we have to do some quick anatomy real fast. We'll come back to this in much more detail later. And this is something that, unfortunately, I guess we didn't emphasize in the last section. So, because again, I've started grading the lab exams, and there's one question in particular that I see that most people have gotten wrong so far that I've seen. Although I'm only about a third of the way through it, so I am hoping that it will get better. So let's put that there, and let's put that there. Now, over here to the side, let's have a chunk of hyaline cartilage. So here is a chunk of cartilage. One of the things that we talked about with cartilage is avascular. All right, doesn't have blood supply directly to it. So if you remember, we talked about what it needs is it needs a layer of tissue on the outside to be able to provide it with the oxygen and the nutrients it needs. Does anybody remember what tissue type that is found on the outer surface surrounding cartilage?
Regular.
No, it's a dense irregular connective tissue. Now, is this the only place? Well, close, Alina. Is this the only place in the body where we find dense irregular connective tissue?
No.
No, of course not. So this dense irregular connective tissue that happens to be wrapped around cartilage, we can give it a name based on its location. And as Alina pointed out, that name of this tissue that wraps around the cartilage is a perichondrium. Any of that sound vaguely familiar?
Mm-hmm.
Excellent. Well, there's something we need to know about bone. Here is, for instance, a cross-section through a long bone. So I got a long bone in my arm, I cut through it, and so it's got the hollow center where we find the bone marrow. Here's the matrix of the bone, and here's the outside. Well, it turns out that bone also has a tissue that wraps around it on the outside. This tissue that wraps around it on the outside, it plays an important, it stabilizes blood vessels, it acts as an attachment point, it provides some protection, and it turns out that a tissue type of it is indeed a dense irregular connective tissue. And since I wrote it off right there to the left, I'm just going to abbreviate it there. However, unlike the one on cartilage, this one is wrapped around bone. And so if the one that wraps around cartilage we call a perichondrium based on its location, guess what we call the one that wraps around bone based on its location? There you go, periosteum. I mention this because deep underneath the periosteum, right up against the bone, is where you find these osteogenic cells. So osteogenic cells can be found right here on the outer surface of the bone underneath the perichondrium. But there's one more thing. It turns out there is yet another dense irregular connective tissue that is located lining the cavity of our bone as well. This is, yet again, tissue type a dense, irregular connective tissue. But this one that is inside of the bone, there we go, is the endosteum. Here's one of the important things to remember about all of these. If I were to take a chunk of this perichondrium, a chunk of this periosteum, a chunk of this endosteum, rip them off and looked at them under the microscope, they would all look the same. They are all dense, irregular connective tissues. The only difference in them is where they are located. And so that is why we give the three of these names based on their location. And the other reason I mention this is because, all right, whoever's flying the helicopter really has to make sure that their microphone is muted, okay? There are also osteogenic cells next to the bone underneath the endosteum as well. So these unipotent stem cells, these osteogenic cells, are located under the periosteum and under the endosteum. So now we'll talk about class in just a minute. We aren't there yet. So our unipotent stem cells are located inside the periosteum and the endosteum. And don't worry, we'll go back to that picture. We're not done drawing on it yet, so don't worry about that.
Inside or underneath them?
Both. If you think about it, underneath them would be kind of inside of them, closer to the bone. So you could describe it either way. All right. So one more piece to the puzzle. Osteogenic cells, as we mentioned, and I'll rewrite it here, are unipotent stem cells. So that means when they divide, they can become one thing and one thing only. And what is that one thing that you think that they become?
Osteoblast.
Osteoblasts, absolutely. There is one thing and one thing they can become. They become our immature bone cells. All righty. Questions on that? Excellent. So then let's talk about osteoblasts. Osteoblasts are immature cells. And as we mentioned, immature cells, were they the ones that made the matrix or are they the ones that maintain the matrix?
Make.
Make, exactly. And in fact, the way I remember is B for blast and B for build. These are the ones that build the matrix. They're the ones that make the matrix. This making of the matrix is a process called osteogenesis. It's kind of a cool process. What we have is our friend, those collagen fibers that we've talked about many, many times. And what put a little blob-like osteoblast here. Osteoblasts take calcium, take the phosphorus, take the magnesium. They take all of those materials out of the interstitial fluid, and they take them and deposit them as a crystal on that collagen matrix. And so they make all these crystals and deposit the crystals on the collagen matrix. And that is how we get that nice crystalline bone material. So again, they calcify the bone. They take the calcium, they take the materials, and they form matrix. And so let's take a close-up look at what we were talking about before. Here is the edge of the bone. And then we have that osteoblast that forms. And as we talked about, that osteoblast is going to start to make matrix. So it starts to deposit some of the calcium on the collagen fibers, and it makes matrix, and then it makes some more matrix, and it makes yet more matrix, and it makes yet more matrix, and more and more and more and more and more, until ultimately the cell has surrounded itself with matrix. Now that it has surrounded itself with matrix, it finds itself in a small hole, a small space in our bone matrix. And if only we had a name for a small space inside of our bone matrix where a cell could live.
Lacuna?
Luna.
Lacuna, excellent. It is now in a lacuna. And if it's in a lacuna, it can't make any more matrix, but it can maintain the matrix that surrounds it. So what's going to happen is the cell matures, and now its job is to maintain the matrix. And when it matures to maintain a matrix, what kind of a cell is it then? Osteocyte. Excellent. It is the osteocytes. Notice one more thing. Remember I said there are 4 different cells that make up our bone tissue. But if you think about it, the first 3 are just the exact same cell at different stages of its life. First as a stem cell, then becomes an immature cell, and then it becomes a mature cell. And it really is that kind of stage. If, for instance, this bone gets broken, and when this bone gets broken, what ends up happening is this osteoblast is, this osteocyte is released from the matrix. Oops, that didn't work hardly at all. That's all right, here, let's you can do it this way. This osteocyte is released from the matrix. It will actually go back to being an osteoblast and will make more matrix again to start the healing process. So it really is kind of just the location of this cell that determines the state of the life that it is in. Now, there is one interesting thing that happens to our osteocytes when they mature. They are locked inside of those caves. Those osteocytes live in the lacunas. And they're surrounded by matrix. That matrix is that crystalline calcium and then the phosphate and all of the other things that are in there as well. And yes, there's water. It is able to get some oxygen and nutrients, but it isn't necessarily going to be able to get all the oxygen and nutrients that it needs. No. And so one of the things that happens is this cell forms these long processes that reach out. And what it's reaching out for is other osteocytes. And where the processes of the 2 osteocytes meet, they will actually form gap junctions. And those gap junctions allow them to pass fluid and nutrients and resources back and forth. So what happens is if I have 2 osteocytes, oh, wrong color. I have 2 osteocytes, and this osteocyte happens to be really close to a blood vessel. It has these long processes that stick out that allow it to, whoops, hold on, go back to purple. These long processes that stick out allow it to get oxygen and nutrients from their blood vessel. And then, because the processes from these form little gap junctions, it is able to pass those nutrients, pass those resources back and forth between the 2 cells. So these osteocytes kind of shrivel up and form these long processes that reach out. And again, I've done a truly amazing job of drawing this, but let's look at the pretty picture from your textbook. Now here's the picture from your textbook, which is okay. I actually like this one a little bit better. I think this one does a better job of showing how this becomes that skinny little cell inside of that lacuna with these big, long processes that stick out. And again, they're the mature cells, so their job is to maintain the bone matrix. Questions on that?
No.
All right, excellent. So that brings us to our fourth cell. Our fourth cell is definitely a horse of a different color. Remember, the first three cells were basically the same cell in different stages of its life. But this third cell, the osteoclast, is actually more closely related to a white blood cell than the osteocyte. And if you notice, if you look at the picture, there's something really interesting about this osteoclast. It undergoes mitosis, but not cytokinesis. Someone interpret for me what that means. That's a big fancy term. That's the kind of thing that you can call grandma on the phone and say, Grandma, guess what? Osteoclasts undergo mitosis, but not cytokinesis. And she's really impressed and she sends you $20 in the mail. But explain to me what that actually means.
It replicates or makes another nucleus but doesn't divide.
Perfect. Mitosis, remember, is the process where it divides the nucleus. Cytokinesis is the process where it divides the cytoplasm, or as Adina pointed out, where the cell actually physically divides. Excellent, perfect, that is correct. So notice what we have here is a cell with multiple nuclei. And notice one other thing about this. This cell, and I'm going to go ahead and use my highlighter to emphasize it on the picture, has this big, huge, ruffled border where it has this massive scalloped edge. And what's the advantage of having this huge scalloped edged or ruffled border?
Increased surface area.
Increases the surface area, absolutely. It increases surface area. What's the advantage of having lots of nuclei? What do nuclei contain? What does the nucleus contain? DNA. DNA is the instruction for making proteins, right? So exactly, this is a cell that produces a massive amount of proteins and has a huge surface area. And the massive amount of proteins it makes are digestive enzymes. And then it has a huge, massive border to be able to make surface contact with the bone so that it can easily break the bone down. All right. These are mostly found in the endosteum. If we cheat and go back to our picture, we can draw one of these things in here. I'll make it green. We haven't used green yet. So again, we're going to have this huge cell with this huge ruffled border and multiple nuclei. And what it's able to do is it is able to release digestive enzymes. And these digestive enzymes break down the matrix. Why might it be useful to break down the matrix?
Is it for energy?
True, but is the energy necessarily, is the matrix, the calcium, those providing energy?
I guess not.
I mean, there is energy in those adipocytes that are in there. That's definitely something that's there. But when we, what's the matrix primarily made out of?
Water and blood.
Water and calcium.
Calcium.
So when we break down the matrix, we release the calcium and the other minerals. from the bone, and we release it into the interstitial fluid. And of course, once it's in the interstitial fluid, where does it go immediately from there? Adina mentioned it, into the blood. So remember, as we talked about, if you have that Diet Coke and a donut for breakfast and your body needs calcium from the bones instead, this is how we get it. We break that down with the osteoclast, and osteoclast, C for clast. And I think C for chew, because that's what they do. They chew up the matrix and they release that calcium into the blood. So it helps to regulate our calcium levels, and we have a fancy name for that. We call it resorption. But here's the other thing too, right? As a baby, you had these tiny little bones with a tiny little cavity in the center of it. And then you grew and got bigger. And as you grew and got bigger, do you necessarily want that tiny little cavity in the center to stay the same size that it was before?
No.
No, we need to get rid of matrix to grow the cavity as well. So how do we get rid of matrix to grow the cavity? Osteoclasts. So notice that's why the osteoclasts are primarily found in the endosteum. Of course, that doesn't mean there aren't any on the outer surface. There are a few, but they are mostly in the endosteum. So our osteoclasts are mostly in the endosteum. All right. Questions on that?
No.
Excellent.
What did you say at the last portion, like the osteoclast, or end up in the endos?
Osteoclasts are mostly found in the endosteum, right?
Oh, okay.
So, yeah. And again, it says that here too. They're mostly found lining the endosteum. Right? There can be some in the periosteum on the outer surface, but they're more on the inside. And again, their job is to break down that bone matrix. And when you break down that bone matrix, you're releasing that calcium into the interstitial fluid, which releases it into the blood. And that process is called resorption. All right. So these help us to remodel our bone, change the shape of our bone as we grow, but dynamically, they also help us to regulate calcium levels in our blood. All right, questions on that?
No.
All right, so those are the 4 cells we find in our bone connective tissue. So let's talk about our matrix. As we know, matrix is made up of fibers and ground substance. And ground substance is mostly water. Again, I know when you hold that skull in your hand, I don't have a real skull or even a pretend skull, I just have a little tiny little crystal skull, right? When you hold that skull in your hand, we think of it as that shriveled up, dried up thing, but bone is very wet tissue. There is 25% water inside of that bone. There is a lot of that in there. And there is 25% protein fibers, primarily collagen. That collagen really serves 2 purposes. The first, as we talked about, is it is the location for the depositing of the minerals in that process of calcification or ossification. But collagen is also what gives, right, bones that little bit of flexibility, that little bit of give. right? You're able to jump up and down and spring and have flexibility and have give to your bones. Again, I can't bend my bone all the way in half. It's not quite that flexibility, but it's not rigid either. We haven't talked yet about the crystals that form our bone, but they're the exact same crystals that form the enamel of your teeth, right? If you feel, and again, technically you're in a lab, you're not allowed to put your fingers in your mouth, right? But if you feel the enamel of your teeth, it's a very hard, rigid structure. Your bone has that same thing. The difference is your bone has collagen, which gives them flexibility, gives them give. So when you jump off your bed onto the floor, right, or you're trying to jump from your bed to the couch, right, to the toilet because the floor is lava, right, and you're making those, that has that flexibility, it has that give, right? On the other hand, if a few months ago, when you found out that poor old Tom Brady was getting kicked out of New England and had to go down to Tampa Bay, and you wanted to drown your sorrow in a big bowl of New England clam chowder, and you bite on that clamshell, while that enamel is very hard, does it have any flexibility? Does it have any give?
No.
No. And so what ends up happening is it can crack. You can break that enamel because it doesn't have that flexibility and it doesn't have that give. And our bones don't start fully mineralized. I made the joke earlier in the semester how you could hold a baby at chest height, drop them, and they would bounce right back into your arms, right? And again, I then warned you, don't drop babies, right? But we also talked about how there's plenty of examples of babies rolling off of coffee tables or rolling off of changing tables, not coffee tables, changing tables. And they typically aren't hurt by that because their bones are mostly collagen. As we age, it becomes more and more calcified, right? So they have less flexibility, less give as we age. And that's why grandma, whose bones are almost fully calcified and has very little collagen left in her bones, when she rolls off the changing table, she doesn't bounce, she shatters. Because of that difference in how much collagen they have in their bones. There's a great way you can see this. We actually have the opportunity to do this in the classroom. We don't have it here. It won't do you any good for this exam, but it can be a fun thing that you can do at home for the semester if you want. And I'll show it to you in a second as soon as we're finished with this. The other half of the bones is our calcium crystals, or more specifically, what are hydroxyapatite crystals. And of course, anything with that many letters in it, you know you're going to have to spell it sometime during the exam. Hydroxyapatite crystals are the hardest substance produced in the body. They form both the enamel of your teeth and the matrix of your bones. It is primarily made up of calcium phosphate, but there's also some calcium carbonate and magnesium and some other minerals that are in it as well. It's actually possible to separate these components of a bone. So the next time you debone a chicken, you can't do this with one that you have already cooked. But the next time you're spatchcocking your chicken or deboning thighs or something like that, I want you to take those two thigh bones. And what I want you to do is clean them as best as you can. And for one of those thigh bones, what you're going to do is you're going to put it in a mason jar, fill that mason jar with vinegar, and put a top on it. And you're going to leave it in there for a month. The other bone, you are going to put on a little, make a little tin foil aluminum tray, put it in your oven, and just bake it in there for about an hour. What you're going to do when you bake that thigh bone for an hour is you are going to dry up all the water that is inside of it, and you are going to destroy all of the collagen proteins inside of it. And what you're going to have at the end of this is a rock hard crystallized bone, which is just those hydroxyapatite crystals. However, that thigh bone that you put into the vinegar, that vinegar breaks down the mineral salts. And so after being in there in a month, what you're going to end up with is a bone that is just collagen fibers. And that bone is going to be all flexible and bendy and twisty, and it is going to be completely mobile because you'll have broken down all the minerals. Like I said, we have several sets of these in the classroom I usually get to share with you. We didn't get the opportunity to do that, but it's something that if you want to amuse yourself with, it's something you can do the next time you have the opportunity. All right, it wasn't worth making you go out and buy thigh legs to do this a month ago so that we could experience it today, but if you're interested, it's something that you can do as a learning exercise. You know, impress your kids and neighbors with it. All right, so as I mentioned, we start with these mostly collagenized bones, and then we have to calcify it, or we have to mineralize it, or we have to ossify it. And like I said, it's those osteoblasts depositing those mineral salts between and onto those collagen fibers that makes our bone. Now, as we also talked about in the previous section, there are actually two specific types of bone connective tissue. Anybody remember what those two specific types were?
Spongy bone.
And?
Compact.
Compact, excellent. Absolutely, that is correct. We have spongy bone and compact bone. So two specific types. Compact bone is the most common, and it is the one that we saw when we looked at our bone connective tissue in the previous section. And remember, as we talked about, the key characteristic of a compact bone is that organizing structure that was the osteon. If you remember, those osteons were made up of rings of matrix around a central canal, the space at the center, and that organizing structure. Oh, and then of course we had all of the, how am I going to do this one? Let's do it. I'll cheat and do it this way. All of those lacunas where the osteocytes lived. All right, it looked a little something like that. That was our osteon. That was our organizing structure. And so that is going to be the key for our recognition of our compact bone. Our cancellous bone or spongy bone, and both are acceptable terms, you may use either of that, do not have osteons. Instead, they have these irregular points and processes. If you take the kitchen sponge or any sponge and look at the cut edge of it, does it have just one repetitive pattern that you see again and again and again?
No.
No, there's just all these different irregular points and processes. So some of them are short and fat, some of them are long, some of them are branched, some of them are not branched. There are all these irregular points and processes. And every single one of these we call a trabecula. So all these irregular points and processes, it doesn't have a uniform shape. The osteon has a uniform shape. A trabecula does not have a uniform shape. It has these irregular points and processes, which makes it look like the end of the sponge. And as was pointed out, whoops, I didn't mean to do that. That's all right. The compact bone is the most common, making up about 80% of the skeleton, including all of the superficial bone. When you look at a skeleton and you see that smooth, white, uniform outer bone surface, that is the compact bone. All the spongy bone is located on the inside. So all of the spongy bone is deep inside of the bones. If your spongy bone is showing, please, heck, if any bone is showing, please see a doctor immediately. All right. Excellent. We can look at some pretty pictures and talk about the anatomy of this. Here we have a nice illustration that does it. And here we see all of those irregular points and processes of the compact bone here. And then, of course, here we see all of the osteons of our compact bone. So compact bone on the outside, spongy bone on the inside. All right, we have a lot of anatomy and a lot of vocabulary to go over for this. So it's a teeny bit early. It is 9 o'clock. We've been talking for an hour. Usually we go a little bit longer before the first break, but this is a good natural stopping point, and we have a lot of vocabulary we're going to hit next. So this is the perfect opportunity to take a quick break, refresh yourself, get something to eat, drink, do whatever, caffeinate, and then we'll be back here in 15 minutes to continue our discussion of the anatomy of this. All right, any questions on that? All right, so we will restart at 9:15, and I will start the recording at that point. All right, see you in 15 minutes. I got a great question during the break that I wanted to address real quickly. And that is the ruffled border of the osteoclast a type of microvilli? And the answer to that is no. Remember, microvilli are basically organelles that stick up off of out of the plasma membrane. They are organelles, right? Same thing as cilia. The only difference is cilia typically are a little longer, and right, cilia are motile in that they move back and forth. What we have here is the case where its plasma membrane has a lot of extensions in it this way. And so it just gives it an increased surface area. So these are not organelles that are sticking off the end of it. These are strictly the enlargement of the plasma membrane to just give it more surface area. All right, excellent. So that was a great question. So I definitely wanted to answer that. Are there any other questions before we get into it? Great question. No, these things are not something that you will be responsible for histologically, other than, as we know, the osteoclast, the osteocytes, because osteocytes are found in the lacunas. But histologically, you won't have to be able to distinguish the osteogenic cells from the osteoblasts from the osteoclasts. Now, is it possible to do that? Yes, absolutely. Well, osteocytes you can ID based on their location, but remember osteoclasts, osteoblasts, and the osteogenic cells are all in the same location underneath the periosteum. So is it possible to differentiate them histologically? Yes, absolutely. Do I expect you to be able to do that? No, I don't. So you don't have to worry about that. All right, great questions. Any others? All right, fun. So let's talk first about our compact bone. Again, as we can see, it is the outside bone, and this is that surface. We see a cross section through a small chunk of the bone, and so that's what we're seeing here. And of course, we see these layers of matrix forming the osteons. What you can see, oops, hold on, let's go back here. Here they've pulled out some of the layers of the osteons so that we can see them, and here we see them again as well. An osteon is made up of these layers of matrix, and we have a fancy name for a layer of matrix. A layer of matrix is known as a lamella, lamella being the singular, lamellae with the E being the plural. And notice, as we can see in this, the collagen fibers are oriented at different orientations within each ring. Again, as we know, the collagen is what gives it that resistance to the shearing forces. And by having the collagen fibers in alternating patterns, basically it makes the bone resistant to those shearing forces. And pretty much any way you stress the bone, it's going to have a little bit of flexibility and it's going to have some give, right? Including twisting forces, as we can see here. Now, these particular lamellae basically form concentric circles around the blood vessels and the nerves that pass through the central canal, and that is actually what forms them. So our osteons are formed by concentric lamellae. It's a little big, so let's make that a little smaller. So osteons are formed by concentric lamellae. So we see those concentric lamellae going around like concentric circles around the central canal and form our osteon. So that is how we make an osteon. Here we see it in a cross section. And grab this and move it up here. We can see the layers of matrix. Again, if we use the highlighter, we can see that. So here is a layer of matrix going all around, and here is a layer of matrix going around, and they form these circular layers. And notice in between the layers is where we find the lacunas. The lacunas are, of course, the spaces where our osteocytes live. But notice something else now about our lacunas. Again, here are two lacunas, lacuna 1 and lacuna 2, where the osteocytes live. But remember, as we talked about, there are going to be extensions of their processes that are going to extend out and meet. Yeah, exactly. These are like rings of a tree trunk, absolutely, around the center of an osteon, these concentric circles. Although we'll talk about the tree trunk rings in just a minute. But notice there are all these dark lines radiating out of the lacunas. Those dark lines are the spaces for the processes. Because after all, if you have an arm that sticks out, you have to have a sleeve that wraps around it. And so these tiny little holes, these tiny little lines that radiate out of the lacunas are what are known as canaliculi. Canaliculi means little canals. And so notice as we look closely at the lacunas, we can see all the lacunas have canaliculi, which are what allow those processes to connect to each other and form those gap junctions. So notice, if I showed you this slide on an exam, there are 3 spaces I could ask you about, right? So on the exam, I could have an arrow, like a nice big red arrow like this one. I could point here and ask you to identify this space, and what would your answer to that be?
Concentric canal.
No, central canal. Central canal, or haversion, because good old Bob haversion was the first one who named it. But central canal is at the center. At the center of all osteons is a central canal. All right. I could have an arrow pointing to one of these large, smaller spaces, and ask you to identify that space. And what would that be?
Lacunae.
That would be the lacuna. And I could have an arrow pointing at these tiny spaces, these lines that radiate out, and what would those be?
Canaliculi. Canaliculi.
Canaliculi.
Canaliculi.
Yeah. And again, I don't care how you pronounce it as long as you spell it correctly. And let's take these questions one step further. What do you find in the central canal? What does the central canal contain?
Minerals. Calcium. Blood.
Close. Blood is closer. Is it just filled with blood? That's just blood pooling?
Oh, water. Is it water?
No, blood vessels. Blood vessels and nerves. The central canals are filled with blood vessels and nerves. What do you find in the space that is the lacuna?
Fibers.
Osteocytes.
Osteocytes. Excellent.
Osteocytes. Okay.
And what do you find in the space that is the canaliculi?
Osteoblast.
Well, not the osteoblast, but the processes, the arm-like processes of the osteocytes. Okay, so these are the 3 spaces that contain 3 different things. All right, questions on this? It gets worse from here. So if this doesn't make sense, let me know now, because we got to move forward.
So what did you say, sir, in the spaces of canaliculi? What do we have?
The processes of the osteocytes. No, great question. These processes are not considered dendrites. Dendrites are, there are dendritic cells. There are obviously dendritic processes that come off of neurons. These processes are not called dendrites. All right. All right, excellent. Now, if you notice when we look at our compact bone, And again, here in the illustration, sometimes it's a little easier to see. Notice we have these concentric circles that are forming an osteon, all wrapped around each other. But notice on the outer surface of the bone is a layer of matrix that goes all the way around. In fact, you could say that this layer of matrix would go, if you extended it, around the entire circumference of the bone. And what do you think we call a layer of matrix that goes around the entire circumference of the bone?
Circumferential lamella.
A circumferential lamella. Excellent.
Circumferential. Yeah.
So we have these circumferential lamellae that go around the entire circumference of the bone. All right. But notice one more thing, right? These osteons are like straws. And if you had a bundle of straws in your hand, there would be gaps, there would be spaces in between them. And is that what we want in our bone? Do we want gaps or spaces in between the layers of our bone?
No.
No. So if you notice, the spaces in between the osteons is formed by these irregular layers of bone. Notice these are long and thin. These layers are shorter and fatter. These layers form more of a triangular shape. Notice there isn't just one general shape to these. These are irregular formed layers that are filling in the space inside the tissue. And so what do you think we call the layers of matrix that fill in the space inside of the tissue? Now, the blender had an answer, but I couldn't quite understand it. What do you think we call these irregular shaped layers of matrix? How many times would you like me to put a box around the correct answer?
Interstellar lamella.
Sponge? Oh, yeah.
Yeah, how about the interstitial lamella? Absolutely, interstitial lamella. So notice compact bone. So our compact bone is formed by all three types of lamella. So all three types of lamellae. Concentric, which forms the osteons. Circumferential, that goes around the outside of the bone. And our interstitial, which fill in the spaces. All three types of lamellae form our compact bone. So compact bone is formed by all three types of lamellae. Questions on that? Excellent. So again, we have those concentric lamellae that form the osteons, circumferential lamellae that go around the entire circumference of the bone, interstitial lamellae that fill the spaces in between. And compact bone has all three types. All right, questions on that? All right, I want you to notice one more thing as well. We know that at the center of our osteons are these central canals. And these central canals run parallel to the osteons, parallel to the length of the bone. And we know that they contain blood vessels and nerves. And they're, again, what we called our central canals. However, if we're going to get blood into and out of these central canals, then that means we are going to need to have some spaces that will allow blood vessels to enter into the bone and move between the layers, between the osteons to finally get to the center of the bone. And so these perpendicular spaces are what are known as the perforating canals. Now notice here on our illustration, they've done them as perfect right angles to each other. Are they always going to be perfect right angles to each other?
No.
No, of course not. In fact, if you were so inclined, and quite frankly, you should be, you can go to our website, and on our website, in the modules for this section, you will notice that there are some bone model pictures. Again, this tends to be a very tactile section. And in this tactile section, we actually have some histology models of the bones. Notice as we look at this one here, there are several things that we are able to see on this. Notice they've shown us the layers of matrix, pulled them out that form an osteon, and you can see the collagen fibers in them going in both directions. These would be the ones that are forming the osteons, and so what type of layers of matrix would this be? If on the exam I had an arrow pointing here, I don't like the little arrow, had an arrow pointing here and asked you to identify that layer of matrix, how would you answer that question?
Lamillae?
Circumferential lamella.
Well, okay, so you get partial credit for lamillae, but these are forming the osteons, so as Allison pointed out, they're the concentric lamillae.
Eccentric.
Notice though, here on the outer surface are some layers that look almost flat because again, we have this wedge of the bone where it's going all the way around. Instead, maybe if we change the view, I think there's a top-down view. Perfect. Notice again from this top-down view, we can very easily see the concentric lamillae, but we also see these that almost look flat that are going around the entire outside. What would they be?
Circumferential.
Circumferential lamillae. And notice if we look close, we can see in spaces like here, there's some irregular layers of matrix, and what would we call that?
Interstitial.
Interstitial lamillae. Don't just say interstitial, but interstitial lamillae. Excellent. All right, there's some, oops. Come on, grab that. Thank you. Here's some more right here, for example, and things along those lines. Notice two more things while we're here. Notice if I flip my arrow around, there is this gray layer on the outer surface of the bone. Any idea what that might be?
Is that the perforated?
Cortex.
Periosteum, excellent. This would be the periosteum. What type of tissue would it be? Dense irregular connective tissue. Don't forget to say connective tissue. Excellent. Conversely, notice there, oops, stop it. There is a gray layer lining the inside of it. What type of tissue is that?
Dense irregular connective tissue.
Dense irregular connective tissue, but what do we call this one based on its location?
Osteum.
Endosteum, excellent. Now, someone was right about something else. Notice for this periosteum, there are these layers of fibers that penetrate into the bone, anchoring that periosteum in place. Remember, one of the things that we said is tendons that attach to the bone are more likely to break the bone than they are to tear off of the bone. And that's because they help to form some of these collagen fibers that penetrate deep into the bone, anchoring that periosteum in place, right? And those are what are known as the perforating fibers, or also known as Sharpie fibers. And of course, why are they called Sharpie fibers?
Because they're sharp.
True, that is because they're sharp, but in this case, I know good old Bob Sharpie was the first person who identified them, and so, of course, planted his flag in. While we're looking at this top-down view, we can look at these dark spaces. What do those represent?
Lacuna.
Lacuna. And notice there are tiny little lines radiating out of them. What would those tiny little lines be?
Canaliculae.
canaliculi, although notice that's probably not the best place to see them. Notice if we go back to here and go here. Oh, this one I like the best. Notice here, they've done a much better job on our model of showing us the big lacunas, but also showing us these big, huge, well, not big, huge, big, large radiating spaces coming out of them. So we see the canaliculi really nice on this. So we see those nicely on that there. And this shows us two more things as well. It does kind of look like a wedge of cake. That is what they've done here. Notice also, and I'll change the color of my, there are these, there are these spaces that are all parallel to each other. What would the spaces represented by the pink arrows be?
Spaces. Is that the
Central canals.
Central canals, excellent. And the spaces indicated by the orange arrows, what would they be?
Perforating canals.
Perforating canals, also known as Volkmann. But again, those terms Haversian and Volkmann are very antiquated terms. They're very rarely used. You'll still occasionally see them in the literature. If you want to memorize them, that's fine. But I think perforating and central canals are better phrases. Notice we can see most of this stuff histologically by looking on a microscope slide, but we have this great histology model that I've spent a fair amount of time on. Do you think this model might be on the exam?
maybe?
Yeah, I think so too. I think that that's not giving away any trade secrets by pointing out that this does a great job of showing this on this particular material. Excellent. So that is our compact bone. Let's talk about our spongy bone. Here we see the spongy bone in the head of our femur and lining the medullary cavity, the space in the center. And as you see, it really does look like a cut sponge. That's where it gets its name, spongy bone, because it looks like it. And notice also, it doesn't have that repetitive pattern like the osteon. So every single one of these irregular points and processes, we just simply refer to as a trabecula. Now, notice as we come up close, and I think the next picture shows it even better. Here we go. Notice when we look up close at a cut trabecula, first we see all the irregular points and processes that are the trabecula. But the trabecula are located in the spongy bone, which is inside. If it's inside the bone, can it be made up of layers of matrix that go around the entire circumference of the bone?
No.
No, of course not. So that means it can't be made of circumferential lamella. But are they also forming these nice perfect circles with a central canal at the center?
Yes.
Really? Show me the central canal.
Oh, no, it's not.
No, it's not. So notice it doesn't use concentric lamellae either. So the trabeculae, the spongy bone, is only made from interstitial lamellae. Only interstitial lamellae, only those irregular layers of matrix form our trabecula. Notice all the rest of the anatomy is still the same. We have lacunas, we have canaliculi. Notice they have cheated by removing the periosteum that would, removing the end osteum that would be on the outer surface. So this would be surrounded by an endosteum. And so notice they show the immature osteogenic cells, and they even show us an osteoclast here on the inner side because they've peeled away. Notice the previous picture. I'm going to go ahead and clear my drawings. Notice here on the previous, whoops, wait, hold on. There we go. Notice this picture in pink, they've kind of left the endosteum in place. And here they've removed the endosteum to show us that interstitial lamellae. And they've done that on our model as well. If we go back to our model and look through the different pictures, perfect. Notice when we look at the inside of our bone, we see in purple here, this purple stuff here is the endosteum lining the surfaces of all these irregular points and processes. But notice they've cut away some of the endosteum to let us see those interstitial layers, those interstitial lamella forming the irregular points and processes. And again, that is, vocabulary is so important in this class. On the exam, if I have an arrow pointing here and I ask you to identify that organizing structure, what is your answer to that question?
Endosteum.
Osteum.
Nope. What is the organizing structure that I'm pointing at? First of all, look, I'm pointing at the yellow thing. If I was pointing, so you guys are correct. If I was pointing at the purple and I asked you to identify the structure based on its location, or the layer or the membrane based on its location, then you would say endosteum. If I asked you to say the tissue type, what would you say?
Is that the compact bone? No.
Dense, irregular,
Dense irregular
connective tissue.
Excellent. Here, I'm pointing at the yellow stuff. Now, ideally, and here, let's cheat. We'll zoom in. We'll zoom in and get a closer, better look. Because that's what it would be like on the exam, after all. We'd get a little bit of a closer, zoomed in look. So with this closer, zoomed in look, I have this picture, this exact picture with this exact arrow. And there are basically at least 3 questions I can ask. So let's do them in order. Identify the tissue, be specific.
Dense, irregular.
Nope. I'm not pointing at the purple anymore. I am pointing at the yellow here. Is it better if I move
the
arrow
endosteum.
here? If I move the arrow here, nope. Identify the tissue, be specific. There you go. That is spongy bone, right? The purple is the endosteum that is covering it, but now I'm pointing at the bone. So that is indeed spongy bone. Identify the organizing structure.
Trabecula.
Trabecula, excellent. Identify the layer of matrix.
Interstitial lamella.
Perfect. So there you go. Notice my arrow didn't move, but 3 different questions I was able to ask about that. So again, as always, my goal isn't be tricky. My goal is to be precise. I am going to give you the words that tells you precisely the piece of information I want. You need to make sure that you give me that specific, precise piece of information. All right?
What was the third question for the interstitial lamella? So you mentioned lamella.
Layer of matrix. Identify
Layer
the
matrix.
specific type of layer of matrix. Okay? Excellent. So that is our spongy bone. And hopefully somebody's got their histology handout. Did we cover everything? I think the only thing that we didn't talk about on the slides that we looked at the model was the Sharpie fibers. Is there anything else on our histology for compact bone and spongy bone that we did not mention? I don't have the handout in front of me. Does anybody have it in front of them? Oh, excellent. Red bone marrow. Where would we find? So if we went back to, no, hold on. We're not, I want to go back. We're cheating a little bit because we haven't talked about the anatomy yet. But if we went back all the way back to this picture, right? I haven't asked you the question yet, but what space is this? This space on the inside, well, this is actually the better question. Identify that space. What is that space that's represented here on this picture? Anybody know?
Medullary cavity.
Medullary cavity, excellent. And what would that space contain?
Bone
Marrow.
Bone marrow, exactly. Now, in the long bone here, it would be primarily, in a mature one, it would be yellow bone marrow. And then notice there's these individual spaces in the spongy bone. These individual spaces between the spongy bone is where you would find the red bone marrow. But we'll talk about that more when we get to our long bone and its anatomy. All right. that is it for the histology. I think that's everything we need for the histology. So what we get to do now is talk gross anatomy. And when we talk about gross anatomy, there are actually 6 structural classifications of our bones. Your book does talk about all 6 of them, but it does tend to emphasize 4 more than the other 2. And there's a good reason for that. So let's identify that. What are the 4 main structural classifications that your textbook emphasizes? Give me one of them.
Long bones.
Long bones, excellent. How do we identify a long bone?
Long bones. They're compact bones.
Not all the way.
That are largest in the body.
True. Yeah, they can be the largest of the body, although there are some very small long bones, right? That bone in the tip of your pinky is a long bone. Is that one of the largest bones of your body?
No.
No, you're right. Definitely some of them, like our femur. Our femur is a long bone. It is the longest bone in the body. How would you generally describe a long bone? The long bone is long?
No.
Okay, excellent. They are narrow bones, so I like that. So again, as someone has pointed out, they are longer than they are wide. We don't want to use long in the definition because long isn't. So they're, let's say, tall, narrow bones. So they're tall, narrow, thin bones. Elongated, typically with enlarged heads. Enlarged ends. Excellent. I know you guys rattled them all off. Give me another type.
Flat.
Flat bones. Excellent. How do we define a flat bone?
It's probably wide and thin.
Excellent. Wide, thin bone. But there's one other key, broad, wide, broad, thin bone. But there's one other key characteristic that our flat bones have. So they're a wide, there we go, they're curved. Excellent. What's next?
Irregular.
Short.
Short.
Excellent, short. How do we define short bones?
Smaller than long.
Yeah, they're small too. They're small, but they're small block shaped. All right, they're kind of cubes, like block shaped, cube shaped. And then I also heard someone say irregular. How do we define irregular bones?
They don't have regular edges.
True, well, that could be the case, although it's not necessarily true for all of them. Really, in this case, this is where we can cheat with our definition. Irregular
Misshapen.
bones are really the bones that are not long, flat, or short. So bones that are not long, flat, or short are typically called irregular bones. Now, these are the 4, what we think of most commonly as the main structural classifications. And that is because most bones of the body, let me rephrase that, most named bones fall into one of these 4 categories. All right. Of course, what's the key word in that sentence?
Most.
Most.
Most, because most means not all. So there are 2 other structural classifications. What are they?
Is it regular?
No, not regular, although that would be a good guess. Usually if we have, long, we have short. If we have irregular, we should have regular. Excellent, there's one, sesamoid. What are sesamoid bones? Well, as the name would indicate, most are small and shaped like a sesame seed. And where do you typically find them?
Maybe in the abdomen, perhaps?
Tendons.
There you go. They form in tendons. Oops, if I spelled tendons correctly. They form in tendons, right? There are over a dozen different locations where they can form in the body. Basically, what happens if a tendon gets stress on it, one of the things that will happen is a bone can form within that tendon that will help to change the angle of that tendon to try to make it a little bit more efficient, to take some of the stress off of the tendon. There are over, like I said, there are over a dozen different locations in the body where you find them. You can find them in the ankle, you can find them in the hip, you can find them in the wrists. Professional bull riders tend to get them on the inside of their thighs from the stress of trying to ride on those bulls. And so everybody gets them in different locations, so they aren't things that typically can be named. However, there are two that pretty much everybody has, and what are they? They happen to be the largest of our sesamoid bones.
Patella.
Patellas, excellent. The two patellas. And what are the patellas commonly known as?
Knee bone.
Kneecap.
Yeah, your kneecap or your knee bone, exactly. Reach down and grab your kneecaps and give them a little wiggle, right? Everybody have two? Is there anybody who doesn't have one? Anyone missing a kneecap? There are some people, there are a few people who don't, that kneecap, the patella bone doesn't fully form. So there are some people who may not have it, right? It's a small percentage, shown a class this small. It's not likely to be the case, but there you go. We have those, all right? So we have those. And what is the last classification?
Sutural.
Say again?
Sutural.
Sutural, yeah.
Sutural.
Sutural, or what are also known as Wormian bones. Sutural or wormial bones are typically about the size of a grain of rice. And where do you think we find these sutural bones?
The skull.
Yeah, these are found in the skull. The joints where the bones of our skull come together are called sutures. And so these bones are actually found in the sutures of the skull. It's the only place you find them, right?
I was going to say the ear.
Well, no, not quite in the ear. There are some bones in your ear, but is there no slide with the bone classifications listed? Yeah, I've got that slide for you in just a second. We're just going through it together. We'll get to that. But so let's say, for instance, this is part of your skull. And what'll happen is the sutures that form are these irregular connections between the bones. So here is bone A, and here is bone B. And so notice, as I were to point at a particular location, even though the joint is irregular, is this A or B? Where my pointer is that part of bone A or part of bone B?
A.
What about here?
B.
What about here?
B.
B. Excellent. What will happen is that inside of these sutures, small bones will form as the sutures are forming. Most of them are the size of a grain of rice, but some of them can be as large as a $50. Now, obviously, this little bone that forms isn't a part of A, isn't a part of B. It is its own individual bone. Now, do all of us get these sutural bones in the same locations? No. So
No.
again, we can't give names to them, but they're definitely there, and they're definitely individual bones. So remember, as we talked about, you have far more than 206 bones in your body. Great question. Now, they're not the fontanelles. The fontanelles are the spaces where the bones haven't fully formed together. So as these sutures of the bones are coming together, those fontanelles close. But you're right, as that fontanelle closes, as that suture forms, that Wormian bone, that sutural bone can form inside of that space. They're typically not associated with traumas. They're typically just more, great question. I don't know if they're quite spot welds. I don't know if they add to the structure. I think it just more, I think it's more just has to do with the bone forming process, that these small individual bones may form in the space on its own as that is ossifying. So I'm not sure if it forms any kind of important function like helping and healing. It definitely doesn't help in healing because they typically form before traumas take place to the skull. And I don't necessarily think that they, especially since they're the size of a grain of rice, they're probably not adding too much to the structural integrity of the skull. But they do form there. All right.
Well, why not call it like for C? Why not just call it bone A? Why is it considered its own bone?
Because it's not a part of A. It is its own separate bone with its own separate, it has its own border. It has its own, it's not like it's a piece of A that is broken off. It is its own piece of bone that formed in that location, independent of A and B. All right. And again, if you don't like my pictures, let's take a look at the pretty pictures from your textbook. Here is our parietal bone, an example of a flat bone. Thin, broad, right, curved. There is a long bone like the humerus. Our sharp bones like the carpals of the wrist. Yeah, page 22 has a good of the atlas. We'll talk about that. Vertebrae is a good example of an irregular bone. Here, so okay, do you like this picture a little better? Notice here, this sutural bone is a completely independent bone that is formed separate from these other two. So it's not a part of this one. It's not a part of that one. It's its own independent bone that is forming in that space. And then we have the sesamoid bones like the patella bones. All right. Questions on this? All right, let's talk about the anatomy of these individual bones, starting first with the long bones. Long bones are found in the appendicular skeleton. In fact, almost all bones of the arms and legs are long bones. What are the only exceptions?
The carpal bones.
Excellent. The kneecap is one exception. The other is the carpal bones, the wrist bones, which are short bones, and what else?
And the feet, we can say the
tarsal bones, exactly. The tarsal bones of the feet, the heel and ankle of it are short bones as well. So we have the short bones, we have the sesamoid bone that is the patella, and we have the wrist bones. Those are the only bones that aren't long bones. All the rest of the bones are long. We have this great picture here, but let's start from scratch with a simple, pretty picture of this. Go ahead and clear that. And talk about a long bone. As we mentioned, a long bone is going to have a long, thin, narrow body, and then it is going to have two enlarged head regions. Of course, in a class like this, we're not going to get away with terms like head and shaft region, or head and body, so we have to use the appropriate terms. What is the appropriate term for the long, narrow shaft of a long bone?
Diaphysis.
Diaphysis. Excellent. Let's change that to that, and let's make it a little smaller so I can fit this all on here. And the name for the heads?
epiphysis.
Epiphysis, excellent. We do have one issue though. We have two epiphyses, ES is the plural, and we need a way to tell them apart.
Proximal and distal.
There you go, because all of our long bones are in our appendages. One end is always going to be closer to the core of the body, and that would be the proximal epiphysis, and the one that is further away would then be the distal epiphysis. Excellent. Excellent. So we have identified the individual regions of these. Oops, didn't want to do that. Get that out of my way. There we go. That's what I wanted. Okay, perfect. The anatomy of the diaphysis and the epiphyses are separate. As we kind of already hinted at, the diaphysis is mostly compact bone. but it is hollow in the center. Okay, that's horrible. So I'm going to cheat and grab that line and bring it a little bit closer. It is hollow in the center. And what did we say we called this hollow space in the center again?
Medullary cavity.
Cavity. Whoops. Whoa. And in a mature adult bone, what would this medullary cavity contain?
Yellow bone marrow?
Yellow bone marrow. As an infant, as an adolescent, all of our bone marrow is red bone marrow. But as we mature, the bone marrow in the diaphyses of our long bones gets converted into yellow bone marrow. Now, as we also saw, and I'll cheat by doing this first with a highlighter, we saw on our model that the inner lining of the medullary cavity is lined with spongy bone. So we do have a thin layer of spongy bone that looks bad. Do that. We do have this thin layer of spongy bone. So like I said, mostly compact bone. I spelled compact wrong, but you get the idea. Thin layer of spongy lines the cavity. But that's not the only thing that lines the cavity. We also know, and I'll change colors now to purple, there is a tissue that lines the inner surface of this medullary cavity. And what would this tissue that lines the cavity be?
Endosteum?
No, that would be the tissue based on its location. What would the tissue type be?
Dense, irregular connective tissue.
Excellent. It is a dense, irregular connective tissue. But you guys are right. Based on location, we call it an endosteum. Again, as a grader, those are the things that are frustrating for me. If I ask that question on the exam and you say endosteum, you clearly have knowledge. You clearly understand the information, but you didn't read the question carefully. If I ask for tissue and you give me endosteum, unfortunately, that's not the right answer, and I can't give you full credit for that. So it is important to read the questions carefully so you can answer them correctly. I say it for every test. I say it numerous times. People often lose points, not because they don't know the information, but because they don't read the questions carefully. You have to read the questions carefully so you can answer them correctly. All right?
Professor?
Endosteum would be the name of the layer or the structure based on its location. And that will be the key word that I use as the prompt on the exam to tell you that. Identify this layer based on its location. Yes, someone said, instructor?
Yeah, I had a question. So you said the thin layer of the spongy lines the cavity. Is that the top part, and then the endosteum is on the inside? Or does the...
Yep, so let's cheat and look at a better illustration than the one I've been drawing. And again, we looked at it from straight on, but let's look at it from the top. Come on. Oh, that's because I'm zoomed in. That's the problem. Hold on. I need to zoom out. Okay, so here we kind of see an example of this here. Notice with our model here, here is the end of the bone on the inside. Notice in gray or purple is that endosteum that is lining the inner surface. So it lines the inner surface and it's over the top of the spongy bone. That's what we saw when we looked at it from the inside. Right now, we are sitting in the medullary cavity, looking towards the outside of the bone. And as we look at the wall of the bone, we see all the irregular points and processes of the spongy bone, all those trabeculae, but they're covered by the purple membrane, which is the endosteum. Now, our model maker has cut some of those endosteum away, but if we were really sitting inside the bone and looking at them, would we see the layers of matrix here? No, the whole thing we would see would be covered with that endosteum, because that endosteum covers all of that spongy bone. So in a real bone, if we were sitting inside of a real medullary cavity looking at the edge, we would see the spongy bone, and the spongy bone would be covered by the endosteum. Does that make sense?
Yes, thank you.
Excellent. All right. Of course, we know this isn't the only dense irregular connective tissue on the bone. We know that there is also, oops, no, there and there. We know that there is a, well, that's way too big. We know that there is a dense irregular connective tissue located on the outer surface of the bone. This is also a dense irregular connective tissue. So again, the tissue type is a dense irregular connective tissue, which I'm abbreviating here because we've already written it out once. And again, based on location, this dense irregular connective tissue, which is on the outside of the bone, we call what?
Periosteum.
Periosteum. There is no difference between the purple layer and the green layer. Both are dense irregular connective tissues. The only difference is their location. One is on the outside of the bone, so that one, its location, we call it a periosteum. One's on the inside of the bone. That location, we call it an endosteum. But both are dense irregular connective tissues. All right. Now, that brings us to our epiphyses. Our epiphyses, unlike our diaphysis, are mostly spongy bone. Right. There is a thin layer of, oops, a thin layer of compact bone on the outer surface of this. Because again, all the bones have to have compact bone on the outer surfaces. Yeah, that was horrible. You get the idea. Thin layer of compact bone. But the majority of the bone on the inside are those irregular points and processes of our spongy bone. Right? Here, those trabeculae formed by the interstitial lamellae allow for multi-axial stress. It helps for compression and for multi-axial stress. So we have this mostly filled with spongy bone. And because it's irregular, there's all these spaces in between. So these spaces are going to be lined with an endosteum. So our spongy bone is lined with an endosteum, and the spaces are going to be filled with red bone marrow. Even as adults, we still need to have the ability to make new bone cells. And so that bone marrow is typically located in the, for the long bones, in the epiphyses. Has anyone here ever donated bone marrow before?
Yes.
Was that an enjoyable experience?
No.
No. First, I want to say thank you for your donation. That is an incredibly generous thing for an individual to do. And one of the reasons why is because it is so incredibly painful. Typically, the largest amount of bone marrow in your body is either in the proximal epiphysis of your femur or in your hip bones. And so what they do is they take a very large bore needle. Sure, they give you a little, subcutaneous anesthetic to try to help, but it doesn't do a very good job. And they take this massive, huge bore needle and penetrate it into the bone and then suck out the bone marrow. And one injection with one needle, are they going to be able to pull out enough bone marrow for a transplant?
Mm-mm.
No. 2, 3, 4. How many times did they inject you to pull out bone marrow? Do you remember? Do you know?
I don't remember. I think it's around 4 or 5.
Yeah, 4 or 5. It is an incredibly painful process. And like I said, it is an incredibly wonderful gift that you did. So again, I commend you for doing that. That is remarkable that you did that. That is amazing. And it is not a pleasant experience. So again, they have to go to these kind of areas to get the bone marrow so that they're able to make those donations. All right. One more thing about the epiphyses. The epiphyses are typically where the bones form their joints to other bones. And so as we know, we want to cover and protect the outer surfaces of the bone where they form those joints. So what type of material do we put on the outer surfaces of our epiphyses to protect the ends of the bone and form those joints?
Cartilage connective tissue?
Yeah, what kind of cartilage? Do you remember?
And
Particular?
Well, you were correct. So absolutely, the tissue type is hyaline cartilage. But it's not the only place we find hyaline cartilage in the body. So based on its location, the fact that it is on the end of the bones for forming the joints, based on that location, we call it articular cartilage. Excellent. Because articulate means to form a joint. So that's just all it is. It's saying this is the cartilage that forms the joint. Excellent. So one more piece to the anatomy, really two. Notice the diaphysis and the epiphyses are dramatically different from each other. And do you think that the bone just instantly transitions from one anatomy to a different anatomy? No. There's going to be, oh, wait, hold on. Oh, well, I didn't really want that to be. No, I do have to fix that. Excellent. So our hyaline cartilage, which based on location is the articular cartilage. Excellent. So there is a transition region, basically from the diaphysis to the epiphysis.
Epiphyseal line?
Well, that's what we'll find in there. You have the right idea. This transition region is what we would commonly refer to as the neck of the bone. But again, we're not going to get away with a term as common as neck. Anybody know what that transition region is referred to as? There we go, metaphysis. And again, we are going to have both a proximal and a distal metaphysis. So proximal epiphysis, proximal metaphysis. That's that transition region. However, as someone mentioned, inside of that transition region is yet another piece of hyaline cartilage. This hyaline cartilage is what we commonly refer to as the growth plate. Of course, we're not going to get away with terms like growth plate on the exam. The appropriate anatomical term has been mentioned a couple times is the epiphyseal plate. Now it's ironic because the epiphyseal plate is located in the metaphysis, but it holds the epiphysis on, and that's why it is called the epiphyseal plate. And as I mentioned, it is hyaline cartilage. Now I mentioned it's the growth plate because in this area, what happens?
Transition?
Well, this is growth. What kind of growth occurs at the growth plate? What kind of growth? Bone grows in width,
Height.
height, or length. Our bone grows in length, right? How many people right now are the exact same size that they were when they were born? Anyone?
No one.
Hopefully not, because I'd feel very bad for your parents if, or your mom. I don't care about your dad, but your mom I'd feel very bad for if you were the size when you were born. No, so we have to make our bones longer. Do they continue to get long for our entire life?
No, it's a stop point.
No, there is a stopping point, and that stopping point is when this epiphyseal plate closes. When we reach puberty, usually somewhere between 18 and 25 years of age, what happens is this epiphyseal plate closes. And when it closes, it basically just becomes a thin line of compact bone. And this thin line of compact bone in the metaphysis is what is known as the epiphyseal line. Now, in my drawing, the way I've drawn it is typically one side of your bone, the growth plate open, and the other side, the growth plate closed. No, of course not. There are going to be, typically both are going to be open and both closed about the same time, right? And that actually brings me back to one of the questions, I think, was it Maxim that answered? Hold on, let me go back. Yeah. Notice I'm drawing this dino bone, right? This generic looking bone when we're looking at this picture. And so again, I've labeled them proximal and distal just because, you know, I chose which side I was going to do for that. When we look at a real long bone in cross section, you're going to be able to recognize that bone, whether it's the femur, whether it's the humerus, whether it's the ulna. And because you know its bone features, are you going to be able to know which end is proximal and which end is distal by looking at each individual bone?
Yes.
Absolutely, right? Because again, not all proximal ends are bigger or all distal ends are smaller. Each bone is different. So because you need to know all the long bones, you're going to recognize the long bones. You will be able to orient the bones in your body. And so you will know which end is proximal, which end is distal, because it'll be an actual bone. It won't be an artificial picture like this one here. So like, for instance, I think that is everything I wanted to do. So let's actually do this with the pretty picture from your textbook that does this. Notice this here is the humerus. And based on this, the fact that it's the humerus, which end is the proximal, which end is the distal? Is the top proximal or is the top distal?
Proximal.
Right, how did you know?
It's written there.
Round.
Yeah, well, that's the one way is that because you could recognize what the two ends of the humerus looks like, but we also have the added advantage of that it says proximal around the big round head, and then it says distal around those big fancy looking articular surfaces. But on the real test, it won't have that label next to it, but you'll absolutely have to be able to distinguish it from that.
Spongy bone.
Well, it's true, but all of them. Notice this one's just been cut in half. This one has spongy bone in it as well. They just didn't cut the cross section through it. So we did the diaphysis, we did the epiphyses, proximal and distal, we did the articular cartilage, we did the metaphyses, we did the epiphyseal plate and the epiphyseal line. I think we've hit everything: endosteum, periosteum, medullary cavity. I think we've hit all of our anatomy. So any questions on the anatomy of our long bone?
So how we recognize or distinguish, as you said, like which one is the distal or proximal, so of each bone, each individual bone?
Because you're going to need to recognize the bones. So for instance, if you go in your module, one of the things I've provided with you are some bone handouts that will help you to be able to do your group presentation. So let's say, for instance, your job as a group is to learn, well, let's not use that one. All right. Here is a humerus. Do you need to be able to tell the difference between the two ends? Do you need to be able to tell which end is proximal and which one is distal?
The one on the left is the proximal.
Correct. All right. Here is a radius. Are the two ends different? Do you need to be able to tell which one's proximal and which one's distal?
If the left
Absolutely.
is and the other one is distal.
Yeah, again, this is proximal and that is distal. Absolutely. So yes,
So because of the capitulum, or because of the head of the ulnar and or humerus or femur, because of the head, we can recognize that this is proximal.
But what you have to realize is every bone is different. One of your responsibilities on this test is to learn the bones and bone features of every single bone. And as you learn the bone and bone features of every single bone, right, do you need to be able to orient that bone how it would be in your body? Do you need to know which end is going to be proximal, which end is going to be distal? Yes. So there is no one rule. Each bone is different, and your response, just like each bone has different bone features, it's not like every single bone has, you know, a head, right? Are there multiple bones that have a head? Yes, absolutely, but not every single bone has a head, so you can't say the head is always proximal because every single bone is different. You're going to just need to brute force learn this material, and then as you can orient a bone in the body, you can tell whether it is proximal or distal. There is no one rule to learn.
Sorry, sir, one other question about the group formation. I see the groups, group 1, 2, 3. So have you recognized, like, did you put the people in the groups?
We will be doing that during the break between lab and lecture. I have not formed the groups yet. We'll be forming the groups today. I did not want to form the groups until everybody was here, although I am a little surprised. We have more participants than we have people enrolled in the class. And I know 2 of those are the interpreters, but we're still over enrolled. So I'm going to see who the extra people are. But anybody who is actually enrolled in the class will be put into a group during the next break, likely in the next break. All right. But we're not quite to that break yet. We have a little bit more material we need to cover first. All right. So we did all of that. We did all of that. Yeah, we'll figure it out. Apparently I'm popular. But yeah, we'll figure it out. No, and a couple people are in twice. So yeah, so we'll make it work. All right. We did everything for the long bone. We did all of that. Excellent. Let's look at the anatomy of the other bones. The good news is the anatomy of the short bones, the flat bones, and the irregular bones is essentially the same, with one very annoying difference. Notice the anatomy of these here, and I'm going to go ahead and draw it on this page, even though it's drawn right here. I want to emphasize this. It has a thin layer of compact bone that is on the top and on the bottom, so surrounding the outer portion. And this thin layer of compact bone we call the cortex. Right? As we've talked about before, this organization, cortex on the outside, medulla on the inside, is something we see a lot. And here we see it in bones. Ironically, in bones, they're separated. The medulla is in the long bones. The cortex is in the short bones, the irregular bones, and the flat bones. But we still have cortex outside, medulla inside. And that's the key to these short, regular, and flat bones. There is no medullary cavity. So with that medullary cavity, without a medullary cavity, instead, the entire inside is filled with those irregular points and processes that are the trabeculae. So we have all these trabeculae forming the inside of these bones. We still have a periosteum on the outside. We still have an endosteum lining the trabeculae on the inside. And all of the spaces in here are going to be filled with what? What are the spaces inside in between the trabeculae filled in?
Irregular.
Well, you're right. The trabeculae are formed by those interstitial lamellae, but so what do we fill? Hold on, let's say be more specific. Fill the spaces between the trabeculae with what? What are they filled with?
Marrow,
Yeah, and what kind?
red,
Yep. Yeah, typically our typically our short, flat, and irregular bones typically have red bone marrow for your entire life. Now, are there some larger of these that over time a little bit of yellow bone marrow may form in there? Yes. Everything in this class is more complicated than we talk about. But for the most part, and the sky is blue answer is the space inside is filled with red bone marrow. And that's it. That is the anatomy of our short bones, our flat bones, and our irregular bones, except for one pesky exception. That one pesky exception involves this structure right here. Now, identify, let's ask the questions. Here I've got, again, this crappy picture I've drawn, but we get the point. What, on the test, I could have this picture, I could have this arrow, and I could ask you for the organizing structure. And what would your answer to that be again?
Trabecula.
Trabecula, excellent. I could ask you for the, oh, actually, let's go back. Well, we can do it in this order. Type of tissue.
Dense, regular.
Nope. And again, I will likely say be specific. That should always tell you to something special. Be specific. What type of tissue is this purple line that I've drawn?
Periosteum.
Nope, spongy bone.
Oh,
All right. I could ask the question, identify the layer of matrix that forms this structure.
interstitial.
And what would that be? Yep, interstitial lamellae. Oops. All right, and that is going to be the same for anything. Again, don't just say interstitial. Make sure you say interstitial lamella. Right, we want to use that word lamella. And that is true for short bones, for flat bones, and irregular bones. However, for some reason, and don't ask me what that reason is, it's primarily just because anatomists hate you, in flat bones, and flat bones only, oops, so I spelled only right. In flat bones only, they give a name to the trabecula based on their location. And what do you think that locational name for the trabeculae found only in flat bones might be? I'll give you a hint.
Definitely.
Diploe. Diploe. Why they're diploe only in flat bones, I don't have a good answer for you. Because the anatomy is the exact same in the short bones, it's the exact same in the regular bones. So it should either be diploe in all the bones or diploe in none of the bones. But as I've mentioned many times, anatomists hate us. So the only reason that they did it in the flat bones only is just to mess with it. So welcome to anatomy and physiology. All right. So if this is a flat bone, there is a last question I could ask, and that would be identify this structure based on its location, and your answer to that would be diploe. All right. Questions on that? All right, excellent. We have gone a little long, but that is okay because we are still doing great on time. So let's talk, did all of that. Excellent. So let's talk about the last thing we have to talk about, which is really a feed into our lab. On your bone and bone features list, you have a list, a study guide that shows you all the material you are going to be responsible for. You are responsible for all 206 named bones. For paired bones, you need to distinguish left versus right. All right? That goes without saying. That is everything you needed to know about everything for those. However, you are also responsible for a specific list of bone features. What do you think a bone feature is?
Whether it's long, it's short.
Attachment points.
True. It's not a trick question. A bone feature is a feature of the bone. All right. Here is my pen. As I hold this pen up to the camera, first I have to be able to see myself on the camera, right? This pen has features to it. It has a clip that allows me to connect it to my shirt. It has a lever that allows me to put the tip with the ink on it. And so when I hold this pen in my hand, there are characteristics of this pen, and those are the bone features. So bone features are a characteristic of the bone. These get all sorts of interesting names based on criteria. As you look at the picture here from your textbook, some of them are big round features that we give fancy names like head, or a condyle, or a ramus, or a facet, or a process. And these names have meaning. A foramen is a hole through a bone. A fossa is an indentation in a bone. A tubercle is a small rough attachment point, whereas a tuberosity is a large rough attachment point. And on the exam, can you just interchange words like tuberosity and tubercle? No. So again, you can brute force memorize this information if you want. But knowing and understanding what these names mean can sometimes help you to decipher. A process is a large piece that sticks out. Like I said, a fossa is an indentation. A foramen is a hole. So knowing these are things that could be helpful to you. Every single bone in your body easily has at least 6 bone features on it. Are you responsible for every single bone feature of every single bone in your body?
Yes.
No. What you have is a precise list of how many bone features you're responsible for. So, for those of you who haven't printed it out, first and foremost, shame on you. But secondly, let's look at the handout from class. Under your modules, under lab handouts, we have bone and bone feature anatomy. Let's start easy with the skull. Here are all of the bones of the skull. You need to be able to identify all of them. If there are 2, you need to be able to tell whether they are left or whether they are right. And just out of curiosity, how many bone features of the frontal bone are you responsible for? 1. Excellent, because someone looked at the next page and saw that there was 1. That's convenient. Frontal sinus. How many bone features of the ethmoid are you responsible for?
7.
7. How many bone features of the vomer bone are you responsible for?
None.
None. Does that mean the vomer doesn't have bone features?
No.
No, of course not. It has bone features, but you're not responsible for them on the exam. Does that mean you don't need to know what the vomer is or where it's found in the skull? What do you think? Do you need to know the vomer?
Yes.
Yes, you need to know all of the named bones in the body. So on a picture of the skull, which conveniently enough I have also provided for you under the modules. So in the modules, again, these are pictures from your textbook, but there are some unlabeled skull pictures, for instance, that you can see here. Conveniently enough, they happen to be color-coded, right? I won't be using skulls that are color-coded on the exam. But anybody know where the vomer is? Well, it's a single bone, an unpelled bone, so not surprisingly, it's on the midline. It actually is this narrow bone right in here. This bone right in here is the vomer bone. So that is the vomer. And in fact, if we cheat, and by cheat I mean look at a different picture, there it is in white. This white bone right here in a sagittal section of the skull, that is our vomer. So if I used a picture of a skull cut in this cross section and had an arrow pointing here, do you need to be able to identify this bone?
Yes.
Absolutely. But do you need to know any of its bone features?
No.
No. All right, so you have a precise list of bone features. Let's go to the whiteboard to finish this off. Go ahead and clear this. So again, we have things you're responsible for. All 206 named bones, all paired bones. You need to distinguish left versus right. And you are responsible for all of the bone features on your list. And again, we want to define that bone feature. It is a characteristic of the bone, on the bone. I know that sounds like a simple, silly definition, but I make a point of emphasizing it because not only are you responsible for bone features, but you're also responsible for skeletal features. If a bone feature is a characteristic of, and let's say it this way, of a single bone, what do you think a skeletal feature is? I like that, characteristic of a skeleton, but we can be a little bit more specific. Is a characteristic that is formed by two or more bones. All right? One of the things I do every morning is when I get up and I get ready, I put a little blush on my nice high cheekbones because I have these beautiful high cheekbones that I got from my grandmother, and I like to accentuate them as much as I possibly can. Of course, is there really a bone in here called cheek? No. Your cheekbone is actually a skeletal feature known as the zygomatic arch. And the zygomatic arch is made by two bones with two pieces that stick out. They're an arch because they come together. So this structure in its entirety is a skeletal feature. Now, if I just have one of those bones by itself, do I have that arch?
No.
No, I have a piece of it, but I don't have the arch. If I have the other one by itself, I don't have the arch. I only get the arch when I put the two of them together. So this is a bone feature of this bone. This is a bone feature of this bone. And when I put those two bone features together, I get an arch. All right? So a skeletal feature is a feature that is formed by two or more bones, period, end of story. So what do you think a skull feature would be then? Well, a skull feature is a skeletal feature on the skull. The skull has a large number of skeletal features. So that zygomatic arch that we talked about, that arch, is actually considered a skull feature because it is one of these features of the skull. So it is a feature. Well, let's change our wording. It is a characteristic of the skull that is made from 2 or more bones. And just out of curiosity, how many skull features are you responsible for?
N.
And I like that. Yeah, absolutely. So as we look at our bone and bone feature list,
14.
excellent. Someone counted quickly. Notice down here, these are skull features. For these skull features, not only do you need to be able to identify the individual skull features, but you also must know the, let's actually switch this back to the whiteboard. So for the skull features and skeletal features, for skull and skeletal features, you also need to know the bones and or bone features that make them. You definitely have to know all the bones. You have to know the bones that make them, but you only need to know the bone features on the list. So for instance, when you learn about the orbit of the eye, you will learn that there are 7 bones that make the orbit of the eye. For that, you just need to know the bones. But that zygomatic arch we pointed at, not only do you need to know that zygomatic arch, but you need to know the name of the bone and bone feature and the name of the bone and bone feature that makes them. So if the bone feature is on the list, you are responsible for it. And the good thing is, that's all you need to know for this exam. Well, plus the joints that we're going to throw in a little bit later. Easy breezy lemon squeezy. All righty. Now, to help you do this, I am going to be forming you into groups. For these groups, again, there's going to be a total of 9 groups, so about 3 people in your group. I am going to be giving you a list of the bones and bone features you are responsible for. All right, so these groups are going to be breaking up. You're all responsible for all this information, but your job is going to be to present these to the class. Now, I've done some things to help you with this. All right, I am going to be adding a discussion board to the website, to our Canvas site, where you guys in a group will be able to, everybody in a group will be put into a group discussion board where you can communicate asynchronously. So again, I encourage you to, we're gonna break into groups today. You're gonna have the rest of the class time today to start working on this material after the break, when we do a little bit more talking. But I'm gonna form you into your groups. You'll be able to talk today, talk about a game plan of how you're gonna do this. But if you wanna share links or information or coordinate what you're doing, you can do that there in the discussion board so that you can communicate asynchronously. So that is fine that way as well. Also, you'll notice under the modules, you will see under the group activities, not only the lists of which bones and bone features you're going to be responsible for. So for instance, if you're in group 1 is going to be responsible for the frontal bone, the ethmoid bone, and the occipital bone, and all of its bone features. Group 2, sphenoid, maxilla, and mandible, and so on and so forth. But the other thing that you will see here in the modules is there are some bone and bone feature images. These are pictures that I just completely ripped off of the interwebs. I went to the almighty Google and got these. Here are some skulls. Here are some different views of the skull. Again, sagittal sections, all of these things, and some disarticulated bones. Let's do that. Let's go back real quickly to our list. If you are, I'll do this on the bone and bone feature list, so you can do this on your bone and bone feature list. Here are the bones of the skull. All of these bones you are responsible for identifying in an intact skull. So in the intact skull, you need to be able to identify all of these bones. However, the ethmoid bone, the sphenoid bone, the temporal bone, the mandibular bone, and the maxillary bone. If you are presenting one of those bones, you are responsible for both presenting them in an intact skull and also dislocated, disarticulated, pulled apart. And that's what you see on our modules handout. So I'll give you a second to write those down or to circle those on your list. And so noticeably, conveniently enough for you, if you happen to be presenting the ethmoid bone, for instance, I have given you some pretty pictures of a disarticulated ethmoid bone. Pretty picture of an ethmoid, pretty picture of a sphenoid. I have made these pictures readily available for you. You are welcome to use these for your presentations. I will tell you right now, none of these pictures are on your exam. I made this list actually after I made the exam for this one. So these were pictures that I got from your atlas or from other sources. So again, the goal isn't to memorize these pictures. These are not the only pictures you are responsible for. And while you're doing your group presentation, if you have a great source for this material and want to use your own pictures and your own handouts, you are welcome to do this. You can be as creative with this as you would like. But I have provided pictures here that you can use, you're welcome to use. And so as a group, what your job is to take these pictures and to label them. So that on the exam, not on the exam, when it's time for the group to present, what you're going to be able to do is you're going to be able to say, this right here is the superior orbital fissure. This right here is the foramen rotundum, and so on and so forth as you go through all of this. You are going to show us these things and identify these things for us from all these points of view. All right, that is going to be your job as a group. Now, notice two things. When you're presenting, you're going to have an image like this on the field of view. So you're going to make it all big. It's going to be showing, and you're going to be a tiny image up in the corner of the screen. Notice most of the time when I'm talking, nobody's looking at me. Everybody's looking at the pictures that we are talking about. And the same thing is going to be happening when you're giving your presentation. Also, you're not being graded on your presentation skills. So if you want to have all of this stuff written down on a piece of paper and you want to sit here and read off that this is the foramen rotundum and this is the foramen ovale, and you want to do that, that is totally fine. You're not being graded on your presentation skills. Do you need to be on screen? Yes. Do you need to present this material? Yes. And you're doing it as a group. So if one person wants to show the images and share their screen, and everybody else wants to turn on their cameras and just go in order and identify the things they want to identify, you're welcome to do that however you want. But your job is going to be to present these bone and bone features to the class. This is a group activity. It is a graded activity. But you're not being graded on your presentation skills. So you can read it. You don't have to look at the screen if you want. You can heck, be turned around backwards. It makes it harder to point at what you're saying. But as long as we can hear you and you're able to point at what you want to point at, as long as we get the information, that is what I care about. I don't care how you present the information. All right? We are going to do this twice. First with the axial skeleton, and then next with the appendicular skeleton. And you'll be using the same groups for this. And these presentations start Monday. So what we're going to do is we're going to take the break. During the break, I'm going to break you up into your groups. Once you get broken up into your groups, we're going to come back and do a little bit more discussion on this. And then I will break you into your break-off groups where you'll be able to talk with your group, make game plans of how you're going to do this. If you want to use the rest of the class time today to start and get it done, you're welcome to do that. What I will warn you right now is that we will be doing this on both Monday and Wednesday, October 5th and October 7th. So on the 5th, we will be doing the skull. Our goal is to get all the way through all of the skull material on Monday. And so then the rest of the axial skeleton, the vertebral column, the ribs and sternum, all of that will be on Wednesday. So you will know once you get a group and once you're assigned your bones, you'll know which day you're presenting on. And we're just going to present in order down the list. So looking at the list, what is the very first bones for the very first group that's presenting? If only, like Laura, I had the list of bones and bone features right in front of me. What's the first one in the list?
Frontal?
There you go. Whoever gets the frontal bone, they're going first. All right. Questions on any of that?
Can you highlight the ones you said that you need to have as intact and not intact?
Yes. So I can go back to that again, and that'll be a good place to actually stop for the break. So let's go back to that list. That was actually it was here. So I'm actually in the right place. But I need to go back to, oh, I gotta first do that. Then I can do that. Bones and bone features. And actually, you know what? I changed my mind. Ethmoid, sphenoid, temporal, mandibular, maxillary, and I'm adding one, palatine. So there you go. Those 6 bones are the bones that you are going to need to be able to recognize and identify disarticulated. Okay? And notice 3 of those, temporal, maxillary, and palatine, are paired bones. So that means for those 3, if I show you a picture of it outside of the skull, you need to be able to tell me whether it's left or right. So remember, when you have those paired bones that you're presenting, remember that is one of the things that you are responsible for. All right, here's what we're going to do. It's going to take me a little bit longer to get the groups formed, and I went a little bit longer with the discussion of everything of this, and we're doing really good on time. So we'll go ahead and take a 20-minute break. That gives you lots of chance to stretch and relax, get something to drink, get something to eat, go to the bathroom, whatever you need to do. And it gives me a chance to get all the groups and everything set up. So we will start back at 11:12. So at 11:12, we will come back. Actually, I'm not going to even bother writing on the screen because I need to stop sharing so that I'm able to break us up into our groups. So 11:12, you got 20 minutes to come back. And then in 20 minutes, we will do the final bit of discussion of what you're going to be doing for this. And then I'll break you into your groups and we will go from there. All right. And just, I'm randomly doing this, so no texting me during the break. I really want to be with Billy. Billy's my favorite. No, I don't care. I'm doing this randomly. This is how it's going to be. All right. Any questions on that? All right. I'll see you in 20 minutes. 11:12. Come back in 20 minutes. before I break you off into your groups, there are a couple more issues I want to talk about, and that's primarily the things that can help you to be successful as you are studying this material. One of those things that can help you to be successful is your practice anatomy lab. If you have not already spent some time looking at this, I want to strongly encourage you to do this. Here under the axial skeleton, you can see some of the anatomy of this. I'm not sure why this is so large on my screen. Hold on, but it's all right. It is a great, great resource. Here, hold on. I don't know what's going on. Some cheat. Let's stop sharing this for a minute. So again, from your study area, practice anatomy lab. Oops, wrong button. Tee. Practice anatomy lab. When you go to your practice anatomy lab, it pops it up as a separate window. There you go. That's the size that I wanted. And you can go to places like the axial skeleton and look at a skull. What's great about this is you can show the labels for many of the features here and hide the labels. You can actually get the opportunity to rotate it. So notice you can change the view of these things, turning it around. You can look at it from the top. You can look at it from the bottom. So it gives you an opportunity to look at this from multiple points of view, the same thing you would get as you are getting comfortable with the material and holding them in your hand. And as you get more comfortable, you can actually take quizzes where it will show you bones, and you have to be able to identify the bones and bone features. Now, when it's doing this type of quiz, is the bone and bone feature it's going to show you always going to be something that's on your list? No, not necessarily. So again, feel free to ignore those and find one instead that you are responsible for, like this one here. And oh, that's clearly the parietal bone. And check your answer. And oh, nuts, I got it wrong. When you're feeling particularly frisky, there is a lab practical that you can take where it'll show you a bone and bone feature, and you actually have to type in the answer and be able to take it that way. So it is a very, very helpful, very useful tool while you are studying that. But what makes this even more powerful is if we go back to our home page, notice there are mobile flashcards. If you click on this mobile flashcard for the human cadaver, for instance, and you could do as much of it as you want, but let's say for starters, I just want to start with the skull. And submit that. And then conveniently enough, I can go through my list and click on every single thing I need to know. I need to know the cribriform plate and the crystal galli. I need to know the ethmoid and the coronal suture and the carotid canal, and on down the entire list of labeling all the things that you want. And once you've done that, you can hit review and it will show you a feature, right? And then you can flip it to see what the correct answer is, or you can go to a quiz that you form based on these flashcards that you make. It is a great, excellent, dynamic tool. These are supposed to be mobile compatible, but right, you also should have your old standby of your photographic atlas. Mine happens to be bound. I think yours is unbound, right? Yours is spiral bound, so you can put it in a notebook, loose. But as you look through it, you get lots of views of a bone like the ethmoid bone or the maxillary bone and the mandibular bone and all the bone features. Again, this is going to show you far more than you're responsible for. So what I encourage you to do is to go in and just highlight the ones you are responsible for. Just highlight the labels of the ones you're responsible for. It'll make this a much faster and easier study guide if you know you can just focus on the things that you are responsible for. You have your textbook, you have your lab manual, you have your almighty atlas, I mean the almighty Google, but your atlas, the PAL CD, things like this, or the personal anatomy lab, practice anatomy lab, are all tools to help you to master this material. All right? Questions on any of that? All right, so everybody was here and I put them into their groups. I've already added those group discussion boards onto Canvas, but what I'm gonna do now is break you into your groups. There are 2 groups that I need to meet with, actually 3 groups that I wanna meet with right off the bat. I'm gonna bounce through the different groups because there's certain pieces of information I want to hold you responsible for. But remember that what you are gonna be responsible for as a group is based on your group number. So again, as we come back here to this page, let me get that out of my way. Go to our modules. Under the group lab activities, skeletal system group presentations, if you are in group 1, you are responsible for the frontal, ethmoid, and occipital bone, right? Group 2, sphenoid, maxilla, mandible, and so on and so forth from all the groups from group 1 to group 8. 8 groups? There should be 9 groups. Uh-oh. Why did I do this? Okay, hold on. This works out perfectly because I noticed one of our groups, one of the people already disappeared. So here we go. Forgot, I used to, in the classroom, we do 9. So apparently I've juggled this here. So this is gonna be easy to fix. Give me 1 second. So there are 8 groups. So you get moved to there, and you get moved to there. Oh, actually, that didn't work. All right. Thanks. Oops. All right, there we go. Now we have 8 groups. So I've had groups. I need to juggle my list a little bit on Canvas. So I'll make that work. So here I have the groups. Once I get you in your groups, stay in your groups till I can get this fixed. And then, but you can start discussing your topics. All right?
I have a question.
I have an answer. What are your questions before we get to the groups?
I don't know if you discussed this, but maybe I didn't hear you. But what was the 30-point skeletal review?
It is a handout that is on Canvas that is not, you are not responsible for yet. I haven't made it available for you yet, but it is a homework assignment that you're going to be able to work on. And actually, that's a great question because that is one more piece that I wanted to add to this, is that one of the things that I encourage you to do in your groups is you're going to be doing these presentations of the bones and bone features, but I do also encourage you in your groups to work on that 30-point review. I'm not giving it to you yet because we need to learn the bones and bone features before you can put them together for the review. But I do encourage you to work on your groups to discuss these and try to come up with the right answers once it is available. However, while I want you to work together, I at the same time also want to ensure that you are writing the answers in your own words. Work on it together, but when it comes to the test, you have to write the answers in your own words. And so I want you doing the same thing here as well. You are going to write your answers in your own words. So discuss it together. Again, some of these answers are going to be 1 word or 2 words. Thoracic vertebrae, right? That obviously you can't write. But if you're writing out a sentence, write that in your own words. I don't want just the same thing repeated again and again. You have to write it in your own words. But again, I purposely have not given it to you yet because I don't want you working on it yet, because you need to learn the bones and bone features first.
Got you.
All right. All right. So I'm going to break you in your groups, start discussing. Please do not leave your groups yet. Yeah, I'd want to at least come to each group and talk to you as a group before you leave. So please do that. And that'll also give me an opportunity to go through and reshuffle the groups so that we have 8 groups instead of 9. So I'm gonna go ahead and open the rooms now so that everybody can then go to their room, and then I will come to your rooms each individually so that we can talk about this material. All right, so there you go, the rooms are open.
Am I crazy or am I the only one that doesn't know what to do now? Are you sending me to where I need to go, or do I have to select where I go?
I should have sent you to, you should be in group 6 is where you should be.
Okay.
Although, hold on, I have too many people in there now. Why, did you leave and come back or something like that?
I did. I had to do a restart. I had a weird notification coming across, so I had to leave
Okay,
and come
so
back.
what I'm gonna do is you were supposed to be in, all right, so I'm gonna juggle these groups a little bit. I'm gonna go ahead and put you into group 3. Let's go ahead and put you into group 3.
Okay.
And Linda,
Yeah.
you also, I think, left and came back, right? Is that what
Yeah.
happened?
By accident, yeah.
Yep, okay, so let's put you in, I'm gonna put you in group 7.
Okay. Thank you.
All right.
I think I left and came back as well.
Daniel, you're supposed to be in group 1.
Okay.
So yeah, you should be able to join that. Here, I'll cheat and do this. Move you there and then move you to there. All right, now you should be able to add group 1.
Okay.
Jade, Maxim, Kyle, Marie, group 3, Allison, Katie, Christopher Alvarez. Group 4, Dina, Allison, Christopher, Kylie. Group 5 should be Angela, Vanessa. Group 6 is now Oxley, Concepcion, Oster, Group 6, Oxley, Oster.
Hey, Professor, can you
Yes.
please help me put me back in my group? My computer went off completely.
What group were you in?
3?
There you go. Seven is 7 and All right, that should work. So,
So
howdy.
sorry. So